Great. So welcome everybody to our latest NISS-Merck meetup. We did our audio test, and today we have three great speakers, which I'll get to in a minute. But before I do, let me just talk a little bit about some housekeeping. If you are having issues or would like to ask a question, the way to do that is to use the chat function. And if you don't have a chat button that's visible to you on your screen, if you look down at the bottom, if you hover over the bottom, you should see a little bubble. And if you click on that, the chat area will appear on your right, and you can type messages to all participants or just to the panel if you're asking a question. Just a minute about NISS. Remember that NISS is a nonprofit National Research Institute for Statistics that promotes statistical research, including postdocs and conferences and independent research. Merck gets involved with NISS through the affiliates program. And upcoming is another webinar, not in this meetup series, but independent of it. What's in a name, data analytics, machine learning, artificial intelligence, and what else? It's all about the intersection between machine learning, artificial intelligence, and statistics and data science. So it's certainly a very important topic these days to statisticians. As you can see on the right, we have a number of great speakers, and that's on September 27th. And if you want more information on that or anything about NISS, you can go to the NISS website, and you can see it, the address at the bottom, www.niss.org. All right. Once again, this meetup is part of a series which we try to do 3 or 4 times a year. I've listed some of the previous ones we've had down the slide there a little bit, and I'll remind you that if you want to catch up on any of those, you can go to the NISS website, and we'll have the slides and a recording posted. Today, we're going to talk about subgroup analysis in drug development, which I think is a pretty hot topic. We have so much data available to us these days in terms of omics and lots of clinical biomarkers and imaging and demographics. We are certainly tempted and interested in trying to use those sorts of data to create subgroups, but as we all know that there are some pluses and minuses in trying to do that, and we will try to look at that in a couple of different ways today. We'll have some sort of background and frame the whole thing. We'll look at it from sort of a regulator's perspective and also a bit from a practitioner's perspective. So with that, we have 3 really excellent speakers today. We have Ilya Lipkovich from Eli Lilly. We have Rob Hemmings, who is now part of a consulting group, Consilium Solomonson and Hemmings. And we have Stephen Ruberg from Analytics Thinking, again, a consulting group. So with that, let's get started with Ilya. He's going to talk about an overview of methods for subgroup identification and clinical trials. Before we get to that, let me give him a little introduction. Ilya is a senior research advisor at Eli Lilly and Company. He's a fellow of the American Statistical Association, has published a number of articles on subgroup identification, but also other areas like missing data and causal inference. He's a co-developer of methods in subgroup identification and also co-author of a book analyzing longitudinal clinical trial data, a practical guide. So with that, let me give Ilya the ball, and we'll move to his presentation, and it's all yours.
You can hear me now?
Yes.
So good morning. Thank you for introduction and for inviting me. So I
We'll get back to your presentation here. Let me get there for you.
Okay, so you have to upload it.
Yep. Okay, you are ready?
Yes.
Okay. It went back to my presentation somehow, so let's try this one more time.
Yeah.
All right, that's yours. You have the ball. You should be seeing your presentation.
It's not yet, but...
Okay. All right, give it a minute. Yeah, you keeps going back to mine, so hang on.
Yeah, I see only yours.
All right, well, let's do this. Let me pass you the ball again, because sometimes that's what gets wrong. So.
Okay. Now it works.
Okay, hold on a second.
Thank you.
Let me give you the ball again. Okay. Can you see it?
Yeah, I can see it now. Yeah.
Okay.
So, hello everyone. So, this presentation, it's a very basic presentation, an overview of methods for subgroup identification in clinical trials. And I also want to acknowledge my long-term collaborator and former colleague, Alex Dmitrienko, with whom I worked on these topics. So I will be talking, start with the basic distinction between predictive and versus prognostic effects. Then I will talk about data-driven versus guidance-driven subgroup analysis, and I'll talk about taxonomy of biomarker identification methods, and I'll briefly give overview of software for subgroup identification, and finish with a summary. So this is a typical set of charts that we present when we want to emphasize the difference between prognostic and predictive biomarkers. So the task of personalized medicine can be translated into statistical language as constructing predictive biomarker signatures that would allow identifying patients with differential treatment response. So here you see several graphs where the y-axis shows some outcome and x-axis continuous biomarker. So the first graph shows biomarker X, which is prognostic as it predicts outcome in the control arm, but it's not predictive. The two lines are parallel. There is no interaction. And X is predictive biomarker in B and C, but in B it's both prognostic and predictive, and in C it's only predictive. And finally, in D, biomarker is neither prognostic nor predictive. Of course, it's a very simple graph, and it's just one biomarker, and the typically challenge is that in multivariate space you have many biomarkers. So, but this is sort of the basic building block. Now, subgroup analysis methods have been around for a while, and there's always a matter of controversy. They are often and rightfully viewed as data dredging that helps sponsors to make substantiated claims about efficacy. Many authors came up with various checklists of principles for subgroup analysis that if you follow the checklist, you wouldn't commit mistake. And so there, for example, Brooks and colleagues 2001 published a list of 25 recommendations with some simulation studies. Rothwell in 2005 proposed a guideline with 21 rules. Sun and colleagues listed existing 7+4 additional criteria for assessing credibility of subgroup analysis. So that perhaps distracted or discouraged many sponsors from subgroup analysis. However, it's interesting that in draft EMA guideline in 2014, there was specifically recognition of issues with current subgroup analysis practices that create disincentive to properly plan the investigation of subgroups. So now there is ideas that, okay, you cannot ignore heterogeneity of treatment effect, and you need to do something. However, how to do it, it's not clear from the guidance, and all guidelines that I ever read on subgroup analysis encourage to exercise caution when conducting subgroup analysis, but it is hard to operationalize because obviously we should exercise caution, but what exactly it means is hard to tell. So here we want to emphasize distinction between data-driven versus guideline-driven approach to subgroup evaluation. So as I try to show, guideline-driven approach perhaps fails to encompass modern scientific approaches to statistical learning and the need for evidence-based personalized, stratified, or precision medicine. The different view is that subgroup identification or analysis can be framed as a special case of model selection. That does make it much more easier, but it connects us with a wealth of literature and research that was done in model selection. So it helps link subgroup identification with other methods, which I'll cover in this presentation. Now here, pre-specified is the entire biomarker subgroup selection strategy, of course, not specific subgroups. So now, what makes data-driven subgroup analysis strategies principled as opposed to data dredging? Well, as they borrow from machine learning methodology, and as you know, there's always bias variance trade-off in applying machine learning methods to the data. You need to exercise complexity control to prevent data overfitting, so that the subgroups that you find in this particular example of model selection would generalize to future data, not just will show in your data. So for this, there are typically there are tuning parameters or hyperparameters that control the search process, and they need to be determined often in a data-driven fashion, for example, using cross-validation. And we know many methods in machine learning, such as penalized regression or shrinking regularization, that have these parameters. Now, evaluating Taiwan error rate for the entire subgroup search strategy, here the key is the entire strategy, as very often we may have multi-stage methods for subgroup identification, and the temptation is only to adjust or to evaluate Taiwan error or false discovery rate associated with later stages of subgroup analysis, ignoring the selection that biomarkers that could have been done at earlier stages. And evaluating Taiwan error can be in general done using resampling, and in some cases there are analytical methods that provide closed form solutions, for example, such as in Lasso. Obtaining honest estimates of treatment effect in subgroups, and that is treatment effect expected in identified subgroups if applied to future studies. And again, it can be done using resampling methods or Bayesian model averaging, empirical based. And again, I emphasize that uncertainty associated with the entire strategy should be accounted for, and not only with perhaps the latest stages. Now, here I give a taxonomy, a very general taxonomy of data-driven methods that appeared in maybe the last 10 years. And we roughly divided into 4 buckets: global outcome modeling, global treatment effect modeling, individual treatment regimes, and local treatment effect modeling. So, we start with global outcome modeling, and global outcome modeling involves estimating outcome function, which is convenient to write as a function of 2 types of variables: the biomarkers X, which is not just single biomarker, of course, it could be high-dimensional set of biomarkers, and treatment, and here we assume treatment just assumes values 0 and 1. Now, the graph on the right illustrates the situation with only single biomarker, which of course is not what we typically have, but it's just used to illustrate the idea. So the idea is that we somehow estimate using different models by arm or joint model including interactions, this function fxt, and typically we use black box methods that gives you non-parametric estimate, though you can use parametric methods as well, and then you can tease out a hypothetical treatment difference from this, which we call delta x, and delta x is just difference for each patient with covariate vector xi. You can imagine the patient was treated and we passed treatment 1, and you get the estimate of the outcome, and then you get estimate for the outcome for that same patient if patient is untreated. And so those are estimated potential outcomes, and for, say, continuous outcome, we can think about treatment effect is summarized by just the difference. And so you estimate this delta i's, which are shown with the red line here on this graph. And now at the second stage of this process, you model this delta x, again, using a second model. So there are two stages here, and it's common for subgroup analysis methods that you don't just have one stage, you have typically multiple stages. That's where the accounting for uncertainty associated with model selection becomes important. Of course, as you can see right from this method, the main challenge for subgroup identification is that you need to somehow estimate the unobservable potential treatment differences at individual level. So here, in a straightforward fashion, we just use the outcome, global outcome model to estimate those, and then tease out those deltas. But those deltas are not observable for any patient in the training data, so those are partially observable because only one treatment is applied for each patient in the parallel clinical trial. And we assume it's randomized trial for now. So some recent methods include virtual twins by Foster, Taylor, and Ruder, and it combines random forest to model the outcome function and CART classification regression trees for further modeling the delta X. Penalized regression adopted for this problem, paper by Imai and Radkovic. Bayesian hierarchical modeling, those by Jones and Extending Dixon and Simon are rather low-dimensional models, but there are high-dimensional models extending Bayesian trees for Henderson and Zhao, so they're non-parametric Bayesian methods. Now, this probably is the most straightforward approach. Now, there are methods that try to obviate the first stage without teaching the global outcome model, and we call it global treatment effect modeling. So instead of fitting this fxt function, it tries to tease out these deltas directly, obviating the need to fit in prognostic effects and get right to predictive effects. And of course, now there are different ideas here. One idea is utilize recursive partitioning methods. And what you do, you try to modify splitting criteria in the tree-based methods by directly incorporating treatment in splitting criteria, so that when you split, you chase the interaction treatment by split interaction, and as a result, you obtain subgroups, so the terminal nodes where the treatment effects differ across the terminal nodes, not the outcome. And there are other parametric methods. Some recent methods, interaction trees by Su and colleagues, GI method by Law and colleagues implemented within GUIDE suite, model-based recursive partitioning by Seibold, which extends the recursive partitioning platform, model-based recursive partitioning implemented in our package. And there is modified covariate method by Shan, and Queen method by Dusseldorf and Mechelin, and some others. Now, as a special case, however, it's becoming extremely important case as modeling individual treatment regimes. Now, this graph on the right illustrates this. So imagine that we don't really try to estimate these deltas for exact deltas for every covariate vector. We just want to know whether it's positive or negative. So if it's positive, we treat with treatment 1 or experimental treatment. If it's negative, that means patients should be treated with control, which is shown here in the green and blue arrows. And perhaps somebody may say that if patient somewhere is close to this indifferent zone, it doesn't really matter what treatment to apply. So maybe you divide data into 3 groups, treat with A, treat with B, and doesn't matter. But the idea is that essentially you need to predict the sign of delta. So that may be easier. And you obviate the need to fit in prognostic main effects, and also you don't really predict the entire difference. You just need to know the sign. So the group of methods emerged that estimate optimal treatment regime by fitting a weighted classifier for treatment as a response with outcome-based weights. Obviously, in randomized clinical trials, we wouldn't be trying to build a model to predict treatment because it's randomized. But the key here is that it's a weighted misclassification loss. It's not just a logistic regression, for example, or tree for predicting treatment. Now we have weights, and weights depend critically on outcomes. So let's say outcome, the larger the better. And okay, patients who did well on their actual treatment would have high cost of misclassification because their outcome is large, and they're likely to have their optimal treatment estimated exactly as the treatment that they received. But patients who did very poorly, for them misclassification loss is small, and they probably would misclassify or classify to the opposite treatment. And so now here also there is a probability of receiving treatment that was actually assigned, which is trivially estimated as a constant in randomized clinical trial. Now in observational data, you can fit a propensity model before applying this method, and that actually then becomes a method for estimating treatment regimes from observational data, which is an extra benefit. Of course, you have to estimate propensity for that and use it in the weight. So recent methods, outcome-weighted learning, all introduced by the groundbreaking paper by Zhao and colleagues in 2012. There are robust kernel methods. There is ROSI method by Xu and colleagues 2015. There are tree and list-based methods that essentially, instead of using smooth biomarker signatures from logistic model, they used recursive partitioning by Zhang, Leiber, and Fu and colleagues. So there's a large number of methods that were developed within this paradigm of optimal treatment regimes. Now, and last but not least, I want to cover the local treatment effect modeling, which is subgroup search per se. And so here we see on the graph on the right, we illustrate the treatment difference, which is, of course, unobservable, but we are not trying even to estimate this treatment difference within entire covariate space. All we care about some kind of bumps or maybe the region when the treatment difference exceeds some cutoff. So this research was motivated largely by bump hunting methods or patient role induction methods by Fisher and Friedman, who argued that if you want to estimate interesting regions in the function, you don't need to estimate the entire function and then chop off this interesting region. Perhaps you can slice it somehow covariate space and get right to the point where, or the region that you are interested in. So I was working in this paradigm with my colleagues, and I already mentioned Alex Dmitrienko. We developed, and other colleagues, we developed several groups of methods, which we call sites family, and also there's method by TSDT by Batu and colleagues, recently implemented as R package. There is a sequential betting implemented also in an R package by Huang and colleagues, and there is a Bayesian model averaging methods, which I essentially consider also these search methods. So I was maintaining for several years the page where I selectively, perhaps with some bias towards methods that I was involved, I listed, some methods that are familiar to me, and I reference R packages, or sometimes the contributors provided the R code, and I put it there. If anybody is interested, send me your code, send me links to your software, and I will be happy to include it. So this is sponsored by a Society for Clinical Trials. And when there are a lot of papers that come out almost every month, it presents you with new novel methods for subgroup identifications. There is much more methods for subgroup identification than subgroups actually found. So when you read it, you kind of question, what can it do for you? How can you trust it? What questions to ask? So here I grouped some questions, and which overlaps with what I listed as features of principal methods for subgroup identification. First question, of course, what's the number of candidate predictors that can be processed in an efficient manner? Some methods just work with one continuous predictor in a very sophisticated way, perhaps fitting some splines. Some methods can handle 20 or maybe 100. Some claim they could do thousands. Now, what's the model space induced by the procedure and how model complexity is controlled to prevent overfitting? As it's a high-dimensional case, we know that any machine learning methods runs challenge of overcoming overfitting. What outputs does the method produce? Is it signature of promising subgroups? Is it personalized treatment contrast for each patient, what you can threshold? Is it optimal treatment assignment? Is it predictive biomarkers ordered by, for example, variable importance or some other score? How the false discovery is controlled, if at all, type 1 error control, false discovery rate? Does the method provide honest estimate, which is point estimate, standard error, confidence interval of treatment effect in identified subgroup that is corrected for over-optimism and can be applied when you plan future study as the basis, for example, for computing power or probability of success? And of course, even if the subgroup is correct, it doesn't necessarily mean that resubstitution method for that subgroup would provide unbiased estimate. Typically, it is biased, and you should use cross-validation or bootstrap or Bayesian methods to adjust for the bias. And this is the table that is updated from recently published tutorial in Statistics in Medicine with Alex Mitrenko and Ralph D'Agostino. Since that time, many new methods emerged, so I updated that table here, and I divided different methods into buckets: global outcome modeling, global treatment effect modeling, optimal treatment regimes, and local modeling. And here, where I could, I provide reference to our packages. And also, we classified it using several features such as modeling type, frequencies, non-parametric, Bayesian, dimensionality, high, medium, large, biomarker selection method. What is the output? Is it signature? Is it predictive score? Is it subgroup? Does it provide control of false positive rate? Does it control complexity? Does it protect you from selection bias when you split the biomarkers with large number of cutoff points? Does it provide honest estimate of treatment effect in identified subgroup? And so we are going to publish this updated table in a book chapter soon. And to summarize, we emphasize here the principle in disciplined use of subgroup identification as opposed to haphazard data dredging. And we treat subgroup identification as a special case of model selection, contrasting data-driven with guideline-driven approach. Unlike standard predictive modeling methods that aim at identifying subgroups with heterogeneous outcome, using methods for tailoring or personalized medicine requires modeling individual treatment differences, which are unobservable, and so that's the challenge. And so we target subgroup with heterogeneous treatment effect, not trying to get just prognostic effects. So that's the challenge. Methods for subgroup identification and analysis borrow from diverse literature in machine learning, multiple testing, and causal inference. That's another challenge. Some methods are rediscovered in different areas. Very often, because it's dispersed in such a heterogeneous set of journals and research communities, sometimes they speak different languages. They don't know much what happens in other communities. So that motivated us to write this overview and tutorial that hopefully was helpful for many people, as at least I got some feedback. A feature of subgroup identification and data mining in general, a recent feature in drug development especially, is the need to control the type 1 error or false discovery rates, which is a relatively new trend in the area of machine learning, as we heard from machine learners for many years that they don't care about significance, and it's all about population, not about hypothesis testing. However, there's increasing convergence between hypothesis testing community and machine learning community. As you know, now there are methods for estimating, for example, the confidence interval from random forest, bagging, and there is a post-selection inference for Lasso, and other penalized regressions. So these communities are converging. Once subgroups have been identified, analysts is facing the challenge of obtaining honest estimate for associated effects that should be expected in the future date, and that's sometimes people forget to do that, but that's an important element of any subgroup search. And with this, I would like to finish my presentation.
Great.
That's some references. Thank you very much.
Thank you, Ilya. I mean, that was just a great introduction and just a lay of the land of all the technical stuff that's going on in this area. So thank you so much for that. And again, if you have questions, we'll try to get to some at the end. So put them in the chat, and we'll do it that way. Okay. So next we have Rob Hemmings. Rob Hemmings is currently works for a consulting firm, as I mentioned before, Consilium, Solomonson, and Hemmings. He was an EU regulator for almost 20 years. He sat on EMA's Committee for Medical Products for Human Use, and for over a decade, and chaired the EMA Scientific Advice Working Party for many years. He's co-author of multiple regulatory guidance documents involving the EMA's guidance on subgroups, so he's certainly a real authority in this area. And but he's now left, as I mentioned, the regulatory network, and is at a consulting firm working in partnership with Thomas Salemson. So with that, let me pass the ball to you, Rob. And
thanks, Dan, for the introduction and for the opportunity to present today. Can I just check that you can still hear me clearly?
Yes, you're coming through loud and clear. Thanks.
Very good. So I still see the final slide of Ilya's presentation.
Yeah, just give it a minute, because on mine it's, in fact, I passed the ball to you. See if you can go up and at that oval near the top, select your presentation now. Do you have the ability to do that?
I think so. How's
Good.
that? What do you see?
I see your slides. Do you see your slides?
I do. That's very
Okay.
good.
Great.
Okay, so thanks again, thanks again, Dan. Yeah, I think I was invited to the meeting today to give some more context to the use of subgroup analyses in regulatory decision making, and perhaps in the context of the EMA guideline on investigation of subgroups, which was actually finalized last year. I think you can find the final version on the EMA website now. So I want to talk generally about analysis and use in decision making of subgroups. So I'll say a little bit about why the interest in subgroups by way of introduction, give some pointers and some signposting around the EMA guidance document, and then distinguish some words on confirmatory subgroups, just picking out one issue as applied to confirmatory or subgroups used in a confirmatory analysis versus the use of subgroup analyses in a more exploratory setting, and finally talk about a third type of subgroup before making some concluding remarks. Why the interest in subgroups? You've heard that people have been writing standards for investigation and interpretation of subgroup analyses over a number of years, and you've seen now from Ilya's presentation both historical and recent advances in methodology for assessment of subgroups. But why are they of particular interest to regulators and regulatory decision making? Well, I've sort of done an extreme dichotomization here. On the one hand, we could ask, well, are regulators and other stakeholders like the health technology bodies in Europe interested in subgroups because they're nasty, or perhaps a better word is conservative, and that the regulators and the other stakeholders see dossiers that are presented from pharmaceutical companies based on data that they've generated and interpreted perhaps in a way that's sometimes a little bit too positive and supports the case that they're trying to make, and so the regulator feels the need to give a critical appraisal and set a high standard for authorization and nitpick over parts of the data which are not so favorable. Or at the other extreme, and perhaps more legitimately in my mind, well, because science demands that we're interested in subgroups. People are different. Disease presents differently within individuals. Various clinical and demographic factors might influence exposure, and individuals might respond differently to treatment even with the same exposure. And really, as scientists and certainly the regulator wants to learn, we want to learn about the effects of medicines and what the effect modifiers might be to a population that's administered a particular treatment. And actually, as a community, when we plan and conduct clinical trials, perhaps we're a little bit disingenuous about this heterogeneity, because on one hand, we strive hard to address it. Perhaps we've gone too far even in terms of extensive inclusion-exclusion criteria, which seek to find a rather homogeneous population to enter our confirmatory studies, not just in terms of the demographic criteria, but in terms of the patients being stable on background medications and not expected to need changes in background medications over the course of a trial, etc., etc. So we try and design away this heterogeneity a little bit. We stratify our randomization in case various factors are prognostic for outcome or predictive of treatment effects. But having recognized that heterogeneity and that potential for differences in treatment effects, we've then historically taken a position that subgroup analyses is dangerous and we want to minimize their conduct and perhaps even ignore their results. And that seems a little bit of a contradiction to me. So I sort of assert that the controversy about subgroups shouldn't be their investigation and the fact that we conduct analyses of subgroups. The controversy is more in how those results and those inferences then go on to be used in decision making. And of course, we have the classic example of which I'm sure you're all familiar, of the ISIS-2 study of aspirin in prevention of further cardiovascular events, myocardial infarctions, where you can analyze that data set and show that 2 star signs, Geminis and Libras, do not benefit from treatment. And of course, that's often pointed to as a warning about the conduct of subgroup analyses. Well, I would see it differently. I don't think it's a bad reason to conduct subgroup analyses, although you can obviously argue about the nonsense of analyzing by star sign anyway. The question would be whether anyone does anything with that result. And if a regulator went on to restrict use of aspirin to the 10 other star signs and ignore the Geminis and Libras, or exclude the Geminis and Libras, that would clearly be a nonsense. So it's not the analysis, it's how the results are used in decision making. So I wanted to present a couple of motivating examples, and these are results that are in the public domain in public assessment reports from applications that have been made in Europe. And the first motivating example is not the conventional analysis, the conventional setting for a subgroup analysis where you have a trial that's overall positive and people are looking around and the company is making explorations to see if any important subgroup has a more neutral or even a negative treatment effect. This is a different setting where the overall results of a trial were not overwhelmingly positive, and the subgroup analysis was actually used to give a positive regulatory opinion. So here we have a trial of Vectibix on top of best supportive care. This was a randomized phase 3 trial in patients with metastatic colorectal cancer, and you can see the results for the primary endpoint on the right-hand side of the screen here. Perhaps a slightly odd Kaplan-Meier curve in terms of its shape, but a clear separation between the treatment arm with the solid line and the control arm with the dotted line. So an overall positive effect. Here, though, you can see subgroup analyses that were performed on KRAS. So this is, let's keep it simple, this is just a biomarker that was dichotomized into patients who had certain mutations and patients who had a so-called wild type KRAS. And this biomarker was related to the mechanism of action of the product. It was on the mechanistic pathway and related to the tumor biology and the way that the mechanism of action for the product would impact on that tumor biology. And you can see on the left-hand side, the patients with the mutated KRAS, no difference at all between drug and control. And you can see on the right-hand side, at least after a period of time when the assessment period would be sensitive, a larger effect than we see in the ITT population. And in fact, the product was then authorized in patients with wild type KRAS on the basis of a subgroup analysis. And why was that still a legitimate decision to take? Well, firstly, because of the strong plausibility that the drug would have a stronger effect in patients with wild type KRAS. And secondly, because there was external evidence from another product with the same mechanism of action where we saw the same pattern of results. And you'll find these messages clear in the EMA guideline on subgroups. When we try and understand effect modifiers and the magnitude of treatment effect or the importance of subgroups in understanding an overall treatment effect, we can't only look at the clinical trial data that are generated. We have to understand the plausibility to see a differential effect in that subgroup and the direction that that differential effect might be. And we should use all available other sources of evidence that might speak to the same question. Anyway, this was an example of a more positive use of a subgroup result. The second example I'm presenting because it reflects evolution in the type of medicines that are being developed and are being authorized, medicines that are targeted to particular excuse me, medicines that are more personalized, more targeted, and for these medicines, perhaps even a stronger rationale to better go and look into and understand results in subgroups. So this particular example was a trial of ipilimumab plus nivolumab. Ipilimumab is a monoclonal antibody that activates the immune system by targeting CTLA-4 and downregulating the immune system. And nivolumab is an anti-PD-1 inhibitor, so another immuno-oncology agent. And this was a trial of the combination versus the two monotherapies. And the mechanism of action for nivolumab was particularly interesting here because there was evidence from some other histologies that the extent of PD-1 expression may be an effect modifier. And in particular, patients with high PD-1 expression may do better on nivolumab than patients with low or no PD-1 expression. And that gives rise to some interesting questions in this study. Even if the overall effect is positive, in patients with high expression, do you need both agents? If you have PD-1 high expression, is nivolumab monotherapy sufficient? If you have low or zero PD-1 expression, is nivolumab effective, or is the best treatment for that particular type of individual only to receive ipilimumab? So even if you have an overall positive result, is there evidence to support the use of the combination of two agents and of course the side effects of two agents across the entire target population. And on the next slide, you can see some of the data from this study. And on the left-hand side, the patients who were regarded as PD-L1 negative, so no expression of the biomarker according to the particular assay that was used. And you can see the combination still remains effective. So that's one question answered. On the right-hand side, you can see the results of the trial by the patients who were regarded as PD-1 positive, where the nivolumab monotherapy seems to perform as well as the combination product, and that's exactly in line with what was plausible before the study, the ipilimumab monotherapy doing worse. And so again, a justification, at least to my mind, of the importance of looking into subgroups in this particular case. Now, I have to give a real health warning around these two examples. I've of course oversimplified them for the purpose of being able to present them in 5 minutes. Yeah, actually the results and what was done with these has a level of complexity that I haven't gone into here. But to paint a picture for you, and in particular where there's targeted therapies and subgroups are defined by a biomarker related to the mechanism of action, a real justification to understand the clinical data in respect of those subgroups. Moving over to talk about the EMA regulatory guidance, it was decided to split subgroups into two types. Subgroups that were used for confirmation and subgroups that were used for exploration. In terms of confirmation, this is addressed through the guideline on multiplicity, and really the question here, or the main topic here is type 1 error control and how the hierarchy, how the confirmatory testing strategy is set up, and so it becomes an issue of multiple testing, and hence is covered in that guideline. On the other hand, you have the more conventional use of subgroup analyses as an exploration that's conducted after the confirmatory test to further learn about the clinical trial data. And that wasn't covered in existing regulatory guidance, and hence the guideline on investigation of subgroups was drafted, draft in about 2014 and final version in about 2018. And as was reflected in Ilya's presentation, when the various papers and publications that have historically tried to talk about when this type of subgroup analysis might be considered sufficiently reliable or credible for decision making, this is a complex topic. This is not only a multiple testing problem anymore. This is both art and science, as you bring the uncertainty that's associated with these analyses into regulatory decision making, and at least to me is more interesting and more fun. Subgroups in some way and effect modifiers are covered in other regulatory guidance documents, of course. But I just wanted to spend a couple of minutes outlining the standards for regulatory decision making. And I've just put on the slide there some criteria for authorizing medicinal products, and these words are taken from European standards. In respect of understanding safety and efficacy, a medicinal product is required to demonstrate therapeutic efficacy and positive risk benefit to be authorized. And that's not translated into whether or not you get a p-value on the primary analysis, the ITT analysis of a clinical trial. It is valid, it is reasonable, it is appropriate for the regulator to consider whether all patients covered by the the target indication, the target population, whether you can justify that you have evidence of efficacy and positive risk-benefit for all patients who are included. Not as individuals, perhaps, but at least all types of patient as defined by the relevant characteristics to do with the disease and to do with the mechanism of action of the product. And I'll come on to talk about that a little bit more. Also, in terms of regulatory standards, I think the big fear and the sort of criticism of bodies who use subgroup analyses in their decision making is that, well, our indication wording will be restricted. Regulators are looking to take patients away from our indication statement. Actually, in my experience, that happened quite rarely. And there's other ways to use information from subgroups in decision making, either to give warnings where there's insufficient evidence to be able to make a conclusion, or to give information to the prescriber about what the effect modifiers might be in order to help in their prescribing decisions, or not to put anything in the product information, but to include discussions and presentations of data in the public assessment report. Of course, the most common use of subgroup analyses in regulatory decision was absolutely nothing. You know, many hundreds of subgroup analyses conducted, even if they showed some evidence of a differential effect, which had no consequence for regulatory decision making, most usually because a differential effect in that subgroup was not plausible and hence was ignored. Just one topic in relation to confirmatory subgroups, and I want to paint a picture by using an example here. Think about a biomarker that's potentially predictive for drug efficacy. So perhaps the higher the biomarker, the more the drug is likely to be effective. And we measure this biomarker on a 0 to 100 scale. So conscious of this, the developer, the sponsor, sets up a hierarchy for their statistical testing. First testing the subset of patients with biomarker more than 50. And if positive, extending the next step in the hierarchy to the patients with biomarker more than 20. And if that's positive, analyze from 0 to 100, analyze everybody. And if that's positive, claim efficacy in everybody. That's a perfectly reasonable hierarchy from a statistical perspective. What it doesn't directly address, however, is the effect in the complementary subgroup. And perhaps what people what sponsors don't realize is how the regulator is interested, or what the regulator is interested in this particular type of hierarchy. So indeed, if the first analysis is positive in patients with biomarker more than 50, that's good. You can still look at the consistency of the effect in that group of 50 to 100. But then the hierarchy proceeds to the next test. And if the test in 20 to 100 or biomarker greater than 20 is positive, the particular regulatory focus there would be, okay, well, I've already known that more than 50, biomarker greater than 50 is positive. So what is that? What is the delta here? What is the effect in those patients that have been added in the subset from 20 to 50? Because actually having a positive result in that second test of patients biomarker more than 20, if that's entirely driven by the patients who have biomarker more than 50 with a neutral effect in 20 to 50, why would you authorize that second group? Certainly you wouldn't authorize the 20 to 50 just because the whole analysis of 20 to 100 is positive. And similarly, you can go to the third step there. So there's regulatory interest in the sort of complementary set. As I say, exploratory subgroups are to my mind, more fun and more interesting. I think there's good scientific justification to try and learn about effect modifiers. Understanding treatment effects in subset of a target population is an entirely legitimate scientific question. But we know that if you conduct multiple subgroup analyses and you look towards the extreme results, they can of course be misleading. And so, as I said before, just as important as the actual data are considerations beyond the trial data, and specifically biological plausibility and replication. And in writing the EMA guideline on this topic, it's positioned as an exercise in signal generation. So investigation of subgroups is positioned as an exercise in signal generation. So it's not the case that if you find something, you have to act on it. The message in the guideline is if you find something in a subgroup, think about it. Think about its plausibility. And I won't go into detail here today, but the guideline separates out 3 different scenarios for the benefit of the reviewer, for the benefit of the assessor, and thinking about judging the impact of subgroups in 3 different contexts. Firstly, where you have overall statistically persuasive evidence and clinically relevant efficacy overall, and you're doing an exploration to understand the consistency of the drug effects. Secondly, where you have overall statistically positive results, but of questionable clinical relevance or a risk-benefit which is borderline or unconvincing, and you're trying to find a subgroup where the effect is stronger to make a positive regulatory decision. And finally, a third scenario where the overall trial data fails to establish statistically persuasive evidence in the full trial population. And there's discussion in the paper around those 3 different scenarios. A really important message, and the one part of it which tries to address Ilya's comment around how to exercise caution, is to recognize that not all subgroups are the same. And when you're setting out to explore subgroups, not all of them are on the same footing. In particular, the plausibility for differential effects in different subgroups. So you might have some key subgroups where the plausibility for a differential effect is higher, perhaps factors that have been used to stratify randomization, evidently factors that are related to the mechanism of action or the pharmacology of the product. And then other factors that might plausibly be predicted for different response, perhaps something to do with the disease, its clinical manifestation or its phenotype or genetic characteristics, tumor biology, etc., etc. And then there's a second set of truly exploratory subgroups where there is no rationale to expect a differential effect. And a little bit like prioritizing endpoints in a study, why do we do that? Why do we put things into families of primary and secondary and tertiary and exploratory endpoints? Well, essentially, it's to control risks of false positive error. And we seek to do, or the guideline seeks to do something different here, to pull out the subgroups where the effect is most plausibly different and focus attention on those, so that extreme findings in subgroups where there's no plausibility for a differential effect can be more easily ignored. The guideline recognizes various tricks that are used by sponsors in discussing subgroups. My interaction test is non-significant, therefore the effects are the same. Well, no, notoriously underpowered. P equals non-significant is no basis to conclude similarity, as we all know. My subgroup wasn't pre-specified, so you should ignore the results. Well, that's the one where, that's the quote that earlier put on the screen. That's when there's a real disincentive to plan properly. If the sponsor can dismiss a finding by simply saying the subgroup wasn't pre-specified, why bother to give this any thought in advance? That's the disincentive part. But you see this argument turned around sometimes. This subgroup was in my list for exploration, and it's the only one where I found a positive result, therefore it has more validity. That's a difficult argument to accept, of course. Arguments around sample size. The study wasn't planned for the investigation of subgroups, so you should ignore all of the results. These are sort of basic and naive arguments which tend to be unhelpful in decision making. Then we have the question of analyzing subgroups on a continuous versus a dichotomous scale. And of course, in terms of exploration, using the full strength of the data and having the subgroup on a continuous scale is a good thing, but often decision making has to be dichotomized to properly inform describers. And maybe in the interest of time, I just jump to the final bullet point there on methods, and these come in various shapes and forms, but I've sort of lumped them together under the term shrinkage. So where we're adjusting the observed point estimate based on usually based on the overall effect size or patterns in other subgroups. And this type of method is compatible with descriptive analyses, perhaps where no differential effect is expected, but not compatible at all with signal generation. And generally, the guidance document discourages these types of methods, preferring to look to the biological plausibility for different effects and whether there's evidence of replication rather than shrinking an observed effect. And I think only very quickly on this third type of subgroup, because historically subgroups have been defined by criteria measured at baseline and discouraged based on events that happen post-treatment. And that's for obvious reasons, because treatment affects the occurrence of events. And so treatment effects that are estimated by comparing such subgroups are biased. And the classic case there is a per protocol analysis, yeah? If we exclude protocol deviators and violators on one arm and compare them to the set of patients who are adherent to the protocol on the other arm, we don't necessarily compare like with like, and we end up with an estimate of the treatment effects that's biased. But actually, this type of subgroup might be really interesting. What's the beneficial treatment effect in the patients who can tolerate the active treatment? What's the treatment effect on duration of tumor response in the patients who achieve tumor shrinkage, etc.? There are some interesting questions that are defined by these post-treatment or post-randomization events. And this is covered in the addendum to the ICHE 9 under the heading of principal stratum, with an open question as to whether methods are available and can be used in regulatory and drug development to give good answers to this type of question. So, science gives us a reason to expect differential treatment effects in terms of the heterogeneity of the patient population and plausibility for inconsistent effects across that population. And that gives a legitimacy to exploration of subgroups, because they give us the potential to learn about treatment. Biological plausibility and replication should have just as much weight in how we interpret subgroups and the consequences for decision making. The sort of basic methods and arguments around subgroup analyses, be aware that regulators are well aware of the tricks, and some of those are listed and discussed in the EMA guideline. New methods are welcome, and I should have said the reason that as with all regulatory guidance documents, the guideline on subgroup focuses on principles and not on methods. And that's because methods develop and methods change. So new methods to investigate and to understand subgroups are always welcome, but not ones that shrink the problem without regard to existing knowledge. And finally, thanks again for the invitation and the opportunity to talk. The methods keep developing, the discussions keep going, and again, that's because this is one of the most interesting problems that we face in drug development and in analysing and interpreting clinical trial data. So I'll conclude there. Thanks, Dan.
Thank you so much, Rob. That was a great list and a great perspective from the regulatory part. So I really thank you for an excellent presentation. Next, we have Steve Ruberg. And Steve, what I'm going to do is I'm going to pass the ball to you, and if you would actually select your slides while I'm introducing you, that seems to work better, because if I select them, it seems to not show up. So Steve received his PhD in biostatistics from the University of Cincinnati and joined the pharmaceutical industry in 1981. He spent many productive years in pharmaceutical R&D, but as well worked in business analytics and areas outside of R&D. After 37 years in the industry, he retired from Lilly as a distinguished research fellow and started up his own consulting firm, which is Analytics Thinking. He's a fellow of the ASA and ISI, has published broadly in statistical and medical journals, and given many presentations at international conferences in those areas. And notably, he was invited to serve as on the advisory board to the Secretary of Health and Human Services in the George W. Bush administration. So thank you for being with us, Steve, and it's all yours.
Okay, Dan, thank you very much. Can you hear me okay?
Yes.
Okay, thanks. So I appreciate being here and the opportunity to speak. Strap on your seatbelts. I'm going to go pretty quickly in order to fit within our time constraints. So here we go. To be or not to be subgroup. First, I have to acknowledge a few people that I worked with at Eli Lilly, Lei Shen, Rick Higgs, and Ilya Lipkovich. They helped shape my thinking in many ways on this particular topic. Although I must say I learned about this topic and became interested in it at about the age of 10 or 12 when I read Alice in Wonderland. And even in the presence of scholarly presentations that we've just heard, I think that maybe a children's novel can teach us something. For example, when Alice meets the Cheshire Cat in Wonderland and doesn't know where to go, she says to the Cheshire Cat, "Would you tell me please which way I ought to go from here?" The Cheshire Cat says, "That depends a good deal on where you want to get to." And Alice says, "I don't care much where," and the Cheshire Cat interrupts, "Then it doesn't matter which way you go." And Alice continues, "So long as I get somewhere." The Cheshire Cat says, "Oh, you're sure to do that if you only walk long enough." Okay. With that, the key messages are be clear on your terminology, be clear on your goals. I'm going to talk about discipline subgroup search. I'm going to advocate for always doing subgroup identification, and then share one last idea with you. Some terminology, precision medicine, target therapeutics versus personalized medicine. Personalized medicine is about you as an individual. Tailored therapeutics is about patients like you, groups of patients who share similar characteristics. And I think we're working in the realm in this webinar around tailored therapeutics and finding those subgroups of patients. Here's a definition that I've used. I can't remember whether I stole it from somewhere or modified it to meet my needs. Three different kinds of tailored therapeutics. The first one is maybe more related to gene therapy or some other areas like that. The last one is around individualized dosing in response of based on a biomarker response with treatment. What Rob just mentioned is post-randomization events. I'd love to talk about that, but time doesn't permit. So I'm going to just focus on this. What most of this webinar is about, subgroups of patients that are expected to have a differential treatment effect. Okay. All right. So targeted therapeutics means finding subgroups in a paper that I wrote with Lei Shen, personalized medicine. We talk about 4 different perspectives on this and how much you know about your biomarker and how that influences the design, analysis, and interpretation of your subgroup findings. And I think Rob touched a little bit on that, for example, with nivolumab and, its PD-L1 kind of characteristics, et cetera, can lead you down a path that's very different than when you're doing maybe something more exploratory. All right, so in clarifying our goals, I think the biggest problem, and the thing that we're all confronted with, and why it makes this problem interesting, or as Rob said, fun or challenging, is to sort out what's real versus what's spurious. And the question I have is, do you want to find a subgroup or not, as indicated in my title? And I think most often, we talk about in terms of biomarkers and tailored therapeutics. The answer is yes. Heterogeneity is there. It's my friend. I want to quantify it. I want to understand it. I want to find the targeted therapeutics. And I think the Vectibix example is a good example of why doing subgroup identification is important. Even if the overall result is positive, it may be driven by a positive subgroup. And I think of my early experiences many years ago with pemetrexed or Alimta, where the non-squamous non-small cell lung cancer patients were driving the overall effect, which was positive, and the drug was approved, but it was all driven by the non-squamous non-small cell lung cancer patients, just as the Vectibix example that Rob showed. But I think there's a thing that's overlooked, and that's when the answer is no. Heterogeneity is my enemy, and I want the treatment effect to be homogeneous across subgroups. And we do these things all the time when we look at certain baseline characteristics like age and gender and race and and geography, and maybe some baseline other covariates like disease etiology or severity, et cetera. And I want to put that in squarely into this conversation as well, because I think it's one that's not addressed, but it's an important one. I'll get to that in section 4. So we advocated in our paper that we published discipline subgroup search. Ilya mentioned that there's lots of people out there with criteria, and I would like to just say disciplined or principled, I think is the word Ilya used, searching for subgroups. It has to be systematic, and it has several characteristics that are distinct from traditional subgroup analysis or exploratory analyses of subgroups. And I think those characteristics, at least my list, is one is pre-specification. Who can argue against that? But pre-specification gives you greater credibility with your results. And there's lots of things to pre-specify in subgroup identification, right? What are you looking for? The list of biomarkers, the covariate space to be searched, complexity of the subgroup definition, all of those different tuning parameters or search algorithms, et cetera. And in that sense, it's no different than any of the pre-specification that's advocated in the ICH-E9 guideline. So that's very, very important in this whole process. The next couple things have been mentioned, adjusting for multiplicity. You know, we have to do something with the significance levels of our findings to adjust for that. And I'm also going to mention some Bayesian thinking a little bit later in the presentation. And then, of course, bias correction. There's many subgroup identification approaches that search for these things, but as Ilya pointed out, there's a bias, an upward bias, due to the selection process, and how you correct for that is very important in understanding and interpreting your findings. That needs to be an important characteristic of any method you utilize. I think your subgroup search has to distinguish between prognostic and predictive biomarkers. I think it has to allow for interactions, that is, not doing one variable at a time, but allow multiple biomarkers to define a subgroup so that there could be interactions not between a treatment and just X1, but a treatment X1 and X2, for example, in your covariate or biomarker space. And then I think another important thing is a kind of I'll call it a partition principle. It allows the identification of a cutoff value for a continuous biomarker. So what level of PD-L1 is the distinguishing characteristic or the appropriate cutoff? And I know Ilya's got some of these features in his table, and I don't know if some of these are new or different or should be added, but that's some of my thinking for what we published as kind of 6 key elements, direct, simple, straightforward, that need to be part of your disciplined subgroup search methodology. Now, with that in mind, I say always do subgroup identification using a disciplined or principled approach. And here's some thoughts around that. So I've heard Sir Richard Pito say, always do subgroup analyses, but never believe them. And I've heard other people tell me that, yes, they've heard Richard Pito say this thing. And it's kind of a little bit discouraging to always do something but never believe in what you're doing. So I'm going to try to give you a path forward where we can maybe overcome that statement. The first is answering this question, do you want to find a subgroup or not? And I'm going to address the less typical case that I'm advocating needs to be rolled into this whole conversation. When the answer is no, and you're looking at routine baseline factors, and you're kind of playing defense, you don't want to find something. You want to avoid trying to explain away that unusual finding when your outcome seems to have some differential response to one of 8, 10, 12, or 15 different baseline characteristics you've identified. I think disciplined subgroup search can be very helpful in this case. As my example here, I look at the results of Toprol-XL in its heart failure study called the MERIT-HF trial. And you'll notice that the US and non-US groups in total mortality showed a dramatic or a very notable difference in response and relative risk. And also down here, males and females were pointed out. And the question is this real or is this spurious? And of course, without any thought or planning or pre-specification or without doing a disciplined subgroup search, you're left with nothing but guesswork and post hoc explanations and justifications, which are rarely satisfying and rarely convincing. And in fact, the label for Toprol has all this vague language that's just not very helpful. You know, the US subgroup and women overall mortality and cardiovascular mortality is less effective. These subgroup analyses are difficult to interpret, and it's not known whether these represent true differences or chance effects. And I think we as statisticians do a disservice to the medical community if we don't think about this and pre-plan it and be disciplined in it, because this label says, gee, women didn't respond as well as men. You go figure it out. We don't know if it's true or not. Somebody go figure it out. And you're left with indecision, confusion, and maybe some women not getting access to a medication that indeed could be very, very useful or helpful to them. There's also the famous example of the Plato trial with ticagrelor and the multi-regional effects, where post hoc these regions were identified and tests were done and showed significant differences, et cetera. And there were many months spent in negotiations and analysis and reanalysis to try to figure out, was this real or not, or how to deal with it, and what the implications were for decision making, as Rob pointed out. So I say always do subgroup identification. What if DSS had been formed for MERIT-HF or ticagrelor, the Plato trial? What if all these factors had been identified? I mean, usually we identify these in a protocol, so why don't we build a pre-specified subgroup identification methodology around them? Say how we're going to adjust the p-values and the estimates. And then we can ask ourselves, would these surprising findings not be surprising anymore? Or if they still held up to be significant with some clinically meaningful effect estimate, then it would cause us to be more certain about diving into a more in-depth medical and scientific assessment of them. All right, so now to the case where you want to find a subgroup, which is the general theme of this webinar and most of the subgroup identification, where you're playing offense and you want to seek or explain heterogeneity amongst patients. And I think using DSS is still very appropriate. The example I've picked here is a recent one, a drug called ramucirumab versus placebo in hepatocellular carcinoma. The so-called REACH trial was published. The overall results were not significant. Ramucirumab versus placebo had a p-value, and overall survival was around 0.15. But when they looked at these groups, subgroups of sex and age and geography, but then also some of these other medical or biological variables, which I've highlighted or blown up here, you can see that there were some differential treatment effects, apparently. Not sure which are real and which are spurious, but the company focused in, and the publication focused in on a biomarker called alpha-fetoprotein, and noting that people that had a level that was above 400 showed a much better result than those that were below 400. Now, how many biomarkers they searched for, not stated in the publication. How they came up with the cutoff of 400, not clearly stated in the publication. But nonetheless, this subgroup was identified. And is it real or spurious? Well, the company took the risk of going forward and doing a second trial called REACH-2, which was ramucirumab versus placebo in the same type of hepatocellular carcinoma patients, but only enrolling patients with this elevated AFP greater than 400. And lo and behold, the treatment effect held up. And actually, if you look back at the last slide, the hazard ratio was 0.67, and the p-value was around 0.01. And now here in the confirmatory trial, if you will, it's hazard ratio 0.71 with a p-value of 0.0199. So a very consistent result. So you say, we've won, the subgroup work panned out. But what if DSS had been formally done in REACH? What if that subgroup, which was defined in the protocol and whatever in advance, what if it had been subjected to a subgroup identification methodology that was pre-specified and adjusted p-values and estimates? And what if they were still significant and meaningful in the REACH trial itself? You say, well, but DSS is hard, figuring this all out and defining this and blah, blah, blah. It's really, really hard. Can we really do it? Well, I say that's the billion-dollar question. The ramucirumab had been approved in the targeted subgroup of high AFP based on the REACH trial alone in 2015 by using DSS instead of doing another trial, spending 3 years, many, many millions of dollars, and tens of thousands of patients not having access to that medication. Ramucirumab was approved by the FDA in May of this year for this indication of hepatocellular carcinoma. So I asked myself, yeah, DSS is hard, but is it $1 billion hard? Could we have been more thoughtful and serious and organized and specified all this stuff and been disciplined in our approach and had a more credible answer with the first trial that may have obviated the need for all this time and money and lack of access for patients with hepatocellular carcinoma, for which there are not very many good treatments. So your run-of-the-mill statistician who's not a knight and doesn't work at Oxford University says always do subgroup identification using DSS so the results are more believable. But one more idea. Suppose there's 100 potential predictive biomarkers that could be important for a new treatment. And you observe, you do 100 tests, you do your experiment clinical trial, you do 100 tests of those, and you find one with a p-value of 0.0001. And even with a very conservative Bonferroni adjustment, that p-value adjusted is 0.01, and you say, Eureka, we've discovered a novel biomarker-defined subgroup. Yay, let's move on, this is great. But I ask, are you sure? Suppose in talking before the study with the scientists and everybody involved, they would have said, gee, this is kind of exploratory. We think there might be some biomarker in this milieu of 100, and the probability that one of those null hypotheses is false is maybe about 20%, based on the biology, the mechanism, the history, other drugs in this class or whatever, and that there's maybe an 80% chance that none of them are predictive. Well, you could assign a uniform prior per biomarker by taking that 0.20 prior divided by 100 so that each individual hypothesis has a prior of 0.022. Okay, now what do we do with that? Well, we're going to do something Bayesian, but a little sidelight here for those who don't know. If you let P0 be the prior probability that H0 is false, that is a biomarker is predicted, and let P be the observed P-value for that test statistic for H0, then we can use Bayes factor to create an upper bound on the posterior probability that H0 is false, as given in this formula here. The upper bound, the posterior probability that the null hypothesis is false, is bounded by this expression here. And if we take that prior of 0.002 and that p-value of 0.0001 and put them into this formula, the Bayesian posterior probability that that biomarker is predictive, that is, that individual null hypothesis is false, it's less than 0.44, less than 44%. Now, you might say that's still reasonably good and worth pursuing, but I would argue that's a very different answer than what many would confer to be a definitive, strong, positive result with an adjusted, the most conservative adjustment possible, p-value of 0.01. I'll note that Berger and Wang and Shen have a much more thorough investigation of Bayesian priors and subgroup identification using this. And in my analytics blog, I just posted something this morning on a drug called dalcetrapib, where I thought there was some misuse of p-values, and a Bayesian approach probably gives a better story. But suffice to say that some people are pursuing dalcetrapib for a very specific minor allele on a gene that is not biologically necessarily related. But there's a trial going on right now to the tune of $150 million in 4 or 5 years, chasing a biomarker that, if you want to read my blog, I explain why I think it's a false positive finding or has a low probability of success. So your run-of-the-mill statistician says, always use Bayesian thinking when doing subgroup identification, so you can quantify how believable the results are. In conclusion, subgroup identification is the holy grail. It's the hardest problem there is because there's all these levels of complexity. I say always do it, whether you're trying to find a subgroup or not. I note again, replication and biological plausibility are very important. And I conclude saying, when I see a significant subgroup finding, I always ask, I wonder what the prior was. And I guarantee you that the prior on Geminis and Libras having, not having some beneficial effect, the prior on that had to be very, very low, like 1 in 1,000 or less, meaning it would have turned out to be meaningless in a Bayesian analysis. And with that, I'll quickly end with thank you.
Thank you so much, Steve. I mean, you have an interesting and provocative point of view, and I think the audience really appreciates that. And certainly a lot of your experience comes into play there. So thanks so much for that presentation. We have had a few questions come in. So the ones that have come in are mostly for Ilya, so let's start there. Ilya, you mentioned a real large group of methods that can be used for identifying subgroups. And each of those methods have different parameters and different assumptions and so forth. The question is has there been any work done to try to comprehensively benchmark those methods against each other in some sort of unified framework, or is that not been done or not even possible?
It's a good question, and there are several papers that compare the selected subgroup identification methods, but typically it's done also by people who propose their own method, and as it happens, they find their method works better than all other methods. So, because people, I think, try to come up with simulation scenarios which may benefit their own methods. But so there is no benchmarking which is comprehensive enough, and I think that's probably one goal that we can do within our subgroup analysis working group. But it's not easy, of course, to do this analysis, simulation studies, and come up with simulation examples that everybody would consider reasonable, because you can come up with a lot of scenarios. And of course, different methods may work better for different type of predictive identifying different type of predictive biomarkers.
Okay, great, thank you. While we're with you, let me ask another question came in for you. This question asked, are there examples where procedures like GUIDE and TSDT have been used to identify subgroups, and then those groups were later tested in phase 2 or phase 3 clinical trials?
If you look at the original papers, for example, for GUIDE, they have examples whether they later confirmed it in future trials. I don't think so. I think it was just confirmed using cross-validation or booster-based method that allowed the authors to construct honest estimates of treatment effect or confidence intervals within them.
Okay.
Dan,
yes.
If I may,
yep, please.
Without giving away too much of my Lilly experience, whatever, I would say more often than not, those methods have been used, or other methods I've seen them used, to basically disqualify or dampen overly enthusiastic scientists or clinicians who want to chase some biomarker because they found a subgroup that has this effect. And when you put it in the context of all the biomarkers and all that kind of stuff, you say, well, it's really not quite as strong as you think in terms of being a definitive predictive biomarker. So I would say more often than not, it's meant to kind of contain enthusiasm or help people understand the likelihood or chances of success going forward, rather than we've identified something that we never thought of or saw before, and now we're going to go forward and test that in phase 3.
Okay, thank you for that. And I think maybe we have time for one more quick question. This one is to Rob. So, and it sort of ties into the question we were just talking about, which is for exploratory biomarkers, something that is found in early clinical or in a clinical study like a phase 2 or phase 3 study. Is it really always the case, if it's exploratory, that we need another study to confirm it, or can the science and the data actually be strong enough that it is believable, and it gets believable enough to have a regulatory or a scientific decision based on that?
Well, thanks, Dan. I mean, in some ways, that's the VectorBix example, yeah. The trial wasn't initially planned to go into the or to support an authorization in the biomarker-defined subset, but the totality of the subgroup analysis data itself, the external evidence from the other program, and the sort of biological plausibility for the stronger drug effect in the wild type subgroup all came together to support an authorization. So possible in principle. And just a short comment to make sure people have seen and understood the parallels between Steve's excellent commentary and the presentation I gave. The defensive scenario, which is scenario 1 in the EMA subgroup guideline, versus the offensive, which is more like scenarios 2 and 3. And of course, when I wasn't when I was a regulator in the European system, I didn't dare to use the word Bayesian for fear of how it might be misunderstood and provoke too much excitement. But when I refer to biological plausibility and replication, I think it's very similar to what Steve is saying of, okay, don't just believe the data. How likely are those data? What do you believe now once you've considered everything, once you considered what you knew before the experiment? It's a very similar message.
Great. Thank you for those comments. And thank you. We're at time, so let's conclude here. But thank you so much to all the speakers. I think it was 3 great presentations, and I'll just mention to the audience that, you know, these guys do this out of just the goodness of their heart. So if you see them at meetings and stuff, buy them a cup of coffee or say thank you in person, because really it is because they donate their time and expertise that we were able to have these sorts of meetups. I'll also give a shout out to Mimi Kim, who really had the idea for this meetup, and always for Christy Trang-Stein, who helps me organize, and Kara Hackman for the technical support. Our next meetup is going to be in January 15th. It's on adaptive designs in the same time period. We hope we can have your attention then as well. And please, if you can, there's a short survey when you disconnect. And certainly if you have topics that you'd like us to cover in the future, please do let us know. So thank you again to the presenters. It was 3 great presentations. I hope everyone has a good rest of the day, and hope to see you next time.
Thank you, Dan.
Thank you, Dan.
Thank you very much, Dan.